Hello friends, welcome to BMS Learning. In this video, we are going to talk about overlapping genes. So what are overlapping genes? Overlapping genes are regions of DNA or RNA that are translated into proteins from two different reading frames to yield two different proteins. As they yield two different proteins, they are also called as dual coding genes. They were first detected in the genome of bacteriophage 5X174. For a long time, they were thought to be mostly restricted to viruses. But computational and experimental studies indicate that they also occur in prokaryotes and eukaryotes. If you noticed clearly, there are two underlined points in the first statement. From two different reading frames and the production of two different proteins. Let's discuss about the first, which talks about reading frames. To understand the concept of overlapping genes, we need to know what are reading frames. In molecular biology, a reading frame is a way of dividing the sequence of nucleotides in a nucleic acid, which can be DNA or RNA molecule. The nucleotides are divided in the form of a set of consecutive and non-overlapping triplets. The concept can be understood properly with the help of a diagram. Suppose this is a stretch of DNA, and these are the nucleotides of which this DNA stretch is formed of. These nucleotides are divided into triplets, and each triplet is represented as a codon. Now concentrate on each codon. Every codon is non-overlapping. That means one codon is not going over the other codon. Now, from reading frame itself, another term comes out, and this is open reading frame. An open reading frame is a part of a reading frame that has the ability to be translated. So, if an ORF can be translated, that means it must have a start codon as well as a stop codon. You can see this in the diagram above. The portion highlighted in the reading frame is actually an open reading frame with both start codon and stop codon. So what about the overlapping genes? When we are talking about overlapping genes, that simply means we are concerned about two different proteins from two different genes. Now the question is, how overlapping genes are doing so? So to get into that, focus on the term overlapping. When two things overlap, part of one thing covers part of the other. Let's understand this with the example of the virus I mentioned in the beginning of the lecture, that is 5X174. The 5X174 is a single-stranded DNA virus and is having several genes named A, B, C, D, and so on. I haven't drawn the way it should be, but for the sake of understanding, we'll take it. So focus on the two genes, A and B. Let's get into this particular portion. Suppose the gene A is formed of this kind of sequence of nucleotides, and we call it as AORA, that is open reading frame A. Similarly, the gene B is formed of this type of sequence of nucleotides, and we call it as BORA, that is open reading frame B. So that means two genes from two open reading frames. Now concentrate. Something interesting is happening here. This particular region of gene A is having these nucleotides. The same nucleotides are present in this region of gene B, which is overlapped by gene A. These genes A and B are referred as overlapping genes. And why we call them as overlapping genes? Because some part of the gene B is overlapped by part of gene A. The important thing is that despite of having similarity in the codon sequences, they produce two different proteins. And this is what the second point we talked about in the beginning. This was all. Thanks for watching.